Okay, recorded. Okay, so hi everyone. We're gonna be doing the postponed week 9 review. Me and Melissa, we're gonna be going over the S phase checkpoints, G2 checkpoint, spindle assembly checkpoint, and yeah, that's about it. And then we'll do questions about the exam and or the problem set afterwards. So before we start, does anyone have any questions? Or should we go right into? Okay. So first slide. Oh.
Oh, sorry.
Okay. Let me actually find my. Okay. So starting off with S phase checkpoint. So pretty much what you want to be thinking about is going off of last week, where we talked about the formation of ORCs, right? Origins of replication. That's where the cell uses to replicate its DNA. And it's important to realize that while that is useful for G1, right? That's where the cell is getting ready to duplicate its double or it's creating the right proteins it needs, it's increasing its size, it's making those ORCs, all that's happening in G1. But when the cell is ready to enter S phase, it needs to kind of do the opposite where it needs to inhibit the formation of new pre-RCs, right? Because if the cell makes new ones, then it'll replicate DNA more than it has to. And that's something the cell really doesn't like. If the cell has either too much DNA or too little or extra duplicated DNA, it's a really bad recipe for disaster where a lot of mutations would occur. So what the cell does is that it uses the formation of pre-RCs that are assembled that are required first, CDKs for their formation, but then after they're created, these CDKs then actually add to inhibit the formation of new pre-RCs. So pretty much what you want to know for S phase is that one of these origin replications, when they are formed, they fire, and that's where the DNA replication can begin. And all these do not happen at the same time. So the cell really needs to regulate when the ORCs form and when they fire. And if we go to the next slide.
I have a question on that
Oh,
slide.
yeah.
As the one that you bold, this complex doesn't require active CDK for formation. I thought it requires active CDK to form pre-IC.
Pre-IC. Oh, are you saying? Oh, the slide says for the pre-RCs. Yeah. So, yeah, CDKs are required for the pre-ICs.
Oh, it does. Okay, I just
Yeah,
didn't.
yeah.
Okay. Yeah. Sorry.
So yeah. So, oh, what happened to my?
Next slide.
Yeah. So, next slide. Oh, we can. Oh, Melissa, could you go to the next slide?
This one?
Oh no, one before. The one before, right
Okay.
before. So continuing for the S phase. So the first step, right, you have your S cyclins activated and they combine with the CDKs, and these inhibit the formation of new PRCs to form. So again, you only want the DNA to replicate once per cycle. It's already done that in the S phase, so you want CDK to then have the new role of then inhibiting future RCs to form, and it does that in 3 ways. One is that it phosphorylates ORCs to prevent the binding of CDC6. Another is that it phosphorylates CD6 itself, which then marks it for degradation via ubiquitin-mediated proteolysis. So it pretty much just sends the CDC to or CDC6 towards the proteasome be degraded. And if this happens, then you want to think about how MCM cannot be recruited. And then the third way that S-cyclin CDKs regulate the formation of new RCs is that it also phosphorylates the MCM, and that pretty much targets it for nuclear export. So those are the 3 ways that it prevents it. It phosphorylates ORCs, phosphorylates the CDC6s and the MCMs. And you want to think about how that's important, that the CDK has the twofold effect, where on the one hand, it's also important to start the formation of ORCs, but when that first initial has happened in the G1 phase, then now in the S phase, then it plays the opposite role. And it actually inhibits the formation of new ORCs. And that also is important to remember, that even though it's CDK that's going throughout every phase of the cell cycle, it really is the cyclins that determine the CDK specific role in the phase it is. So for the next slide, so what happens is that, again, in S phase, you have these cohesion rings that form around the replicating DNA. And these cohesion rings, they pretty much, like you see on the screen, they just hold the DNA strands together throughout and after duplication into the M phase. And it's really important that this happens at the S phase, right when the cell, or right when the DNA is duplicated, because then the DNA strands, the sister chromatids, as you can see in the diagram here, they're already lined up and side by side. So it's really important for cohesion to get on them as soon as it can. And we'll go into later how that's actually a very important step for the spindle assembly checkpoint. But going forward with this, the cohesion rings, they prevent chromosome segregation happening too early, and they also must be present again in S phase in order to assure that the DNA is properly aligned for when they do separate in mitosis. And so at the end of the S phase checkpoint, there's 4 things that the cell wants to make sure. It wants to make sure that there's no DNA damage, right? And it wants to make sure that DNA is replicated completely. Those are the 2 big things, because again, an unequal amount of DNA, or DNA that's too much, too little, is very disastrous. And it also wants to make sure that P53 activation can happen in this phase, and that replication can be stopped at this checkpoint if necessary. And this is a big step also, because the cell really wants to make sure it takes its time to make sure that all of the DNA integrity is intact, and it won't progress farther into this into the next phase, into G2, unless all these are checked. And the importance for P53 is that P53, remember, is a signal for programmed cell death. So it's really important that the cell can switch that on if it needs to, if it detects any DNA damage. But also it's important to remember that if the cell pauses at this checkpoint for too long, then it'll automatically go to cell program death anyway. So that's the 2 things. It wants to make sure that DNA damage, or DNA has its integrity, and if it doesn't, that it can use P53, or if it's in that checkpoint for too long, then it'll just go straight to program cell death. So, and now for the G2 phase. So pretty much for G2, again, you want to remember that the cell is growing again, right? It's replicated its DNA, it's increased its size, it's made all the proteins that it needs to, and now it needs to prep the chromosomes for segregation. So remember, in S phase, the main goal was to make sure that the DNA replicates once and only once, and it's in full integrity. Now for G2, the goal is that the chromosomes are going to be prepped for separation. And also, of course, make sure that the DNA is not damaged. So pretty much, this is where we have our activation of M phase CDKs. And there's another point of confusion for a lot of people. Even though it's the G2 phase, it is the M cyclin that's activated. And without that, then the cell can't go into M phase. So for the next slide, we have the beginning of the G2 phase to the G2 checkpoint. So for the first, as soon as it goes into the G2 phase, M cyclin CDK's levels are rising, but they're still inactive, right? So you want to remember that for the G2 phase, M cyclin levels need to be increased, but they're not necessarily activated. And that's the other difference, that we need to remember, that just because M cyclin binds, or any cyclin binds to a CDK, that doesn't necessarily mean the CDK is active. It might also need to be phosphorylated or have some other inhibitory kinase, which we see here. So pretty much what happens is that CAC phosphorylates the M cyclin CDK with two kinases. One is activating and one is an inhibitory phosphate or inhibitory kinase. So you have, as you see here, the activating kinase CAC and then the inhibitory WEE1. And so what happens is that this is pretty much important for the cell to kind of pause and make sure that the CDK doesn't start prematurely, right? Because remember, we're still in G2, we're not quite in M phase. So it's going to put that WEE1 on to make sure that the cell has time to, again, check for DNA damage, check for proper cell growth and all that. So again, the main point is that M cyclin levels increase. Because of that, CAC and WEE1 are attached to the CDK M cyclin complex, and the WEE1 pretty much holds it on pause until the cell is ready for M phase. So for the next slide, the late G2 phase. All right, this is after the checkpoint. Everything is good to go. Now the M cyclin CDKs are activated. So, and this is done by CDC25, which is an activating phosphatase. Remember that just because you're adding a phosphate to a group doesn't necessarily mean that it's activating it. And this is an example of the very same opposite, where it's actually phosphatase, right? It takes away a phosphate group from the WEE1, which then activates. And this is an example of positive feedback, right? So when this happens, then the steps after that are rapidly increased. So yeah, so that's pretty much that. And by the phosphorylation or phosphatase of WEE1, it takes off that from the complex and this fully activates it so that M cyclin CDK is ready and the cell is ready to go into M phase.
Okay, so I'm doing M phase, and bear with me here because I was sick, so I didn't go to every class this week, so I apologize. So an overview of M phase, it goes from prophase to prometaphase to metaphase to anaphase, and then telophase and cytokinesis. So at prophase, just a reminder, MCDK is active. There's a breakdown of the nuclear envelope so that the chromosomes can be condensed and they can be accessed by the microtubules. And then in prometaphase, microtubules grow and shrink towards their respective locations, and they're looking for the proper kinetochore attachments on the chromosomes. In metaphase, the chromosomes align along the metaphase plate. They have the proper attachments, and this is where the spindle assembly checkpoint occurs. And then once we get to anaphase, this is where the chromosomes that have been duplicated are separated. And then telophase and cytokinesis, the nuclear envelope begins to reform as the cell divides and the chromosomes condense back into the nucleus. So in prophase, like we said, the MCDKs are active and they are phosphorylating the lamins, which break down the nuclear lamina, and that allows for the nuclear envelope to break down. And then they phosphorylate condensin, which induces chromosome condensation. And it's sort of like they're going from long things of spaghetti to shorter, thick sausages that look like X's. And it phosphorylates various maps and motors to facilitate the mitotic spindle assembly. And then in the process of prophase, there is sequential phosphorylation. And this is where phosphorylation needs to happen in a certain order for, for prophase to occur. So I do want to take this one. I honestly don't know what this is from, since I think I missed this.
No, you got it. It was so pretty much sequential phosphorylation. It's pretty much the idea that we want to keep in mind that there is like an important sequence to these things in the cell, right? Especially with CDKs, since they are the master cell cycle regulators. they're the priming kinase for a lot of the downstream kinases. And because of that, inhibiting a CDK will prevent the entire cascade. And that's an example on the left, like we talked about, for P for PI1 for it to be activated, it has to be phosphorylated out of its inactive state by the substrate, which can only happen if the CDK first primes it. So does that make sense? There's a sequence where if you want to activate an inactive kinase, you first want to go all the way up the chain to the CDK for all the downstream effects to happen. So what the important thing is that, say if anything happens to the CDK up top, say it's upregulated or downregulated, and that's going to affect the whole downstream cascade.
Okay, thank you. Okay, so moving on to spindle assembly, which occurs in the prometaphase. And so this is when the microtubules are reassembling into spindles, and so that they can separate the chromosomes during the beginning of M phase. So there's different types of microtubules involved. There's aster, which grows from the spindle pole here and attaches to the cell cortex. And then there's the kinetochore, which grows from the spindle poles as well. Those are shown in blue and attaches to the kinetochores from each of the sister chromatids right here. And then there's the interpolar, which grows from the spindle poles and overlaps at the middle, right here, shown in pink. And so these all work together to help the division. And so the kinetochore is an important multi-protein complex that's built on the centromere of each sister chromatid. So the centromere region is part of the chromosome, and then the kinetochore is a separate protein complex. So there's about 80 different proteins involved in this, and this is where the spindle microtubules are able to attach to each specific chromosome so that they can be separated. And so those were these kinetochore microtubules shown in blue here. And so the way that this separation of sister chromatids happens is through the opposing forces of the motor proteins that we've been talking about. So it's like a push-pull force going on. So the spindle formation requires opposing forces, the kinesins and the dyneins. So they walk towards opposite ends of the microtubule to create opposing pulling forces, which stabilize the spindle so that it's able to pull the chromatids apart. So EG5 is an example of a motor protein that binds to the interpolar microtubules that overlap. And this is plasmin directed, and it pushes spindle poles closer together. Does anybody have any questions on this? I feel like I didn't. Okay, moving forward. So a continuation of this mechanism. This, I honestly, Syed, do you want to take this one?
Yeah. So
Sorry.
pretty much no, you're good. So pretty much for the push-pull mechanism, you want to think about how the cell has to align these chromosomes in order, right? Or align them properly with the sister chromatids. And it does that by balancing tension on push and pull mechanisms. So pretty much what you're gonna have, if I remember this correctly, dyneins that are minus N directed. And then you're gonna have what's the other?
Kinesins.
You're gonna have minus N directed, and you're gonna have positive directed. And so pretty much what those are gonna do is that they're gonna be pushing and pulling along the microtubules and the interpolar microtubule, or the so pretty much that is going to create tension, and that's going to pretty much hold the chromosomes in place in the middle.
Okay.
If that makes sense. Do people have questions? I know that's a piece where a lot of people get confused.
Yeah, I don't really know how to like phrase this question other than really like, I'm just confused about how we know whether it's a push or a pull depending on like where it is in the cell.
Oh, right. So, let's see. Oh, so pretty much as far as the pull, you want to think about how dyneins, right, they're minus N directed. So they're going to be walking towards the minus end of the microtubule, which is going to be towards the pole, right? Towards the edges. And it's going to, like, it's a tug of war. It's going to pull the microtubules away. And then for the push, I'd have to get back to you. Melissa, do you know?
No, I'm sorry.
She doesn't care. Like she doesn't care whether you say pull or push. She could say the binding is pushing it out, or you could say is pulling it out. Like she doesn't care about push or pull. Like you could use them like either one.
Interchangeably.
Yeah.
Okay, that makes sense. Thank you. I think I just got a little bit confused about like the directions and things, but I think I got it now. Thanks.
So moving on to metaphase. When we enter metaphase, the sister chromatids are still secured by these cohesion rings, and they're attached to the microtubules at the kinetochore. And there's the presence of the equal and opposite pulling forces, which was the push-pull, which causes them to line up along the metaphase plate at the center of the cell. And so from metaphase to anaphase, this is where the spindle assembly checkpoint is, which ensures that all of the sister chromatids are properly aligned along the metaphase plate, and that the spindles are attached properly to the kinetochore, so that when anaphase does occur, one chromosome non-disjunction doesn't happen and one chromosome is not. So all the chromosomes get separated. So the cell will inhibit anaphase unless there's proper microtubule attachments at all of the kinetochores. Otherwise, like I said, non-disjunction will occur. So this right here is the process.
What is non-disjuncture?
So non-disjunction is when both of the sister chromatids will go to one side of the cell. So let me find a thing that has a good visual of it. So it would be if this whole thing was pulled by, just say if this kinetochore microtubule wasn't attached and only this one was, so then the entire replicated chromosome would go to one side of the cell, and then it would end up in one replicated cell. So the one cell that's replicated would have double as much DNA, and then the other cell that was replicated over here would have not enough DNA.
Thanks.
Does that make sense?
Yeah, thank you.
Okay. So this is the MAD-BUB CDC20 complex. So the MAD and the BUB are two proteins which sense the microtubule tension on the kinetochore. And so if there's equal tension between the two kinetochores on either side, so think of this as like a mirror image over here. If there's equal tension, then CDC20 is released. And I believe that here, this says it more in general. I mean, more in depth. So once CDC20 is released, it can bind and activate the APC. And the APC is a ubiquitin ligase. And this creates the CDC20 APC complex, which targets the protein securin for ubiquitination. So securin is bound. Where is it? So this is securin, and it's bound to separase right here in blue. And it inhibits the separase's function when it's bound. So this presence of the active APC complex causes here the securin to be degraded by ubiquitination. And so once it's degraded, separase is able to have a conformational change, and it has become activated. And separase cleaves the cohesion that surrounds the two sister chromatids so that anaphase can occur and the separation of the two sister chromatids can occur. And then this complex also targets M cyclin for degradation, so that the M cyclin CDK activity will start to drop. So is there any questions about this part of the checkpoint specifically?
Yeah, I have a quick question. So you just said that APC, active APC will also target MCDK, like to degrade it?
M cyclin.
M cyclin. Okay. And then another thing was, so CDC20 is kind of like released by like MAD and BUB sensing
Yeah.
the tension.
So over here, this is right at the centromere and the kinetochore. So when Mad and Bub sense equal tension on both sides, that will signal. So it's like this is one complex. And when Mad and Bub are sensing equal tension on both sides, is it, wait, say it, is it equal tension on both sides, or is it just tension that it's attached to the microtubule? I feel like I always get that confused.
Oh no, it is I think it is equal tension.
Okay. So when they're sensing this equal tension, then CDC gets released from the complex. And so then it's just available in the cell floating around.
Yeah.
And so then it finds the APC and activates it.
Yeah, because if there's not equal, then you increase the chance that one gets pulled to one
One-
side more,
yeah,
and
and
then
we'll all
that just gets.
Thank
I have a
you.
question about M cyclin levels. Is it true that an increase in M cyclin levels from G1 to M is controlled at the transcriptional level?
A transcription level of.
Of M cyclin.
Yeah, I don't know. Sorry, it's a question from the problem set. I know it's not exactly this one. It's just like a question about M cyclin in general.
Okay. Oh, if it's with the problems, that we could do that after.
Oh, okay.
You want to say?
So moving on to anaphase. There's anaphase A and there's anaphase B. In anaphase A, the chromosomes are pulled poleward, so towards the spindle poles. The chromosomes are walked by the negative directed motors. And the kinetochore microtubules that are attached here are shortened, so they go from being in the center at the metaphase plate, and they're being shortened towards the poles. And then on anaphase B, there's a sliding force, and the spindles lengthen, so the microtubules grow on their plus end. So anaphase A is the kinetochore microtubules, and then anaphase B, I believe, is these microtubules here. And so the spindles lengthen and they're growing on the plus end, which causes the poles to be pushed apart from each other. And this sliding force between interpolar microtubules is from the opposite poles. And so this pulling force is directly on the poles to pull them apart. Does anybody have any questions on that? Okay. And then the last phase is telophase and cytokinesis. And here's,
Sorry,
oh,
I don't mean to interrupt. Can you just sit on that slide for
Okay.
2 seconds?
Yeah.
Thank you so much. Okay, I'm all set. Thank you.
telophase and cytokinesis. So the sister chromatids are in the opposite poles, and now a cleavage furrow will begin to form. And it's created by this contractile ring, which is made of actin and myosin. This creates an indentation in the cell's membrane that will deepen until it hits the spindle that is left in between the two nuclei, and then it'll eventually split into two separate cells. And so remember that because M cyclin CDK activity has dropped because it was being targeted by this active APC complex for ubiquitination, the spindle has begun to disassemble. The chromosomes are decondensing. They're no longer in the thick sausages, and they're back in like the long spaghetti. And then the nuclear envelope begins to reform around the DNA. So the nucleus is reformed in the cell. It's no longer floating around. And so these are the main ideas of the cell cycle that we just want you guys to remember, is that the cell cycle events have to happen in a strict sequential order from G1 to S phase to G2 and to M phase. The cell cycle events involve cyclical activation and deactivation of proteins, which is often by means of phosphorylation with ATP within the cell. And the CDK must be bound to cyclin and phosphorylated in order to be activated. The CDK regulates various proteins in different phases of the cell cycle. Different phases of the cell cycle have their own cyclins. Each cyclin is created, degraded in a particular part and phase of the cell cycle. And regulation for proper function requires activation and deactivation and inhibition. Do you have anything to add, Said? Is that
No.
okay? Any questions, guys? Okay. Thank you for coming. I'm going to stop sharing and then stop recording so that we can